So nitrogen is a principal macroelement that is embedded in all vital structures. And therefore it represents a growth-limiting factor for plants. Since the Green Revolution, inorganic forms of nitrogen, such as ammonia or nitrate, have been intensively used in the traditional synthetic fertilizers. However, the overuse of these inorganic nitrogen-based fertilizers can result in very severe and harmful environmental consequences. On the other hand, organic nitrogen was shown to have a great positive impact on plant growth and especially on the root branching, but the regulation behind these growth-promoting effects remain unexplored. In our project, we are focusing on the role of organic nitrogen in the very early stages of lateral root development. For that, we are using Arabidopsis thaliana as a model plant in two complementary approaches. In the first approach, we are using a special fluorescent line of Arabidopsis that visualizes very specific cells in very early stages of this lateral root organogenesis. We will then apply organic electronic technique that enables us to deliver different nitrogen compounds with very high spatial resolution, and therefore we can only target these specific cells with different nitrogen treatments. These cells will be then isolated using fluorescent-based cell sorting technique, and then will be used to investigate the differential gene expression as well as metabolite profilings in response to different nitrogen conditions. This approach will help us to identify cell-specific novel genes and molecular players that are involved in the growth-promoting effects of organic nitrogen. The second approach is based on our preliminary data, and it suggests the interplay of organic nitrogen with plant hormones, such as auxins and cytokinins, that are another group of plant regulators, plant growth regulators that are very well known and already very intensively studied for their role in root development. Our pilot studies suggest that organic nitrogen modulates the levels and metabolite profiles of both these hormonal groups, and therefore we want to focus also on the identifying of hormone-related genes that can be involved in the regulation of root system architecture in response to organic nitrogen. So taken together, the gained knowledge of molecular basis of organic nitrogen mode of action can help us to identify novel genes and molecular players that can be manipulated to bioengineer the important forest and agriculture plants with stable, rich and uptake-efficient root system, as well as to develop novel organic nitrogen-based fertilizers that contributes to the recent sustainable strategy of agriculture and forestry. Thank you.
Thank you.